All right, I think we just start. So hello, good afternoon, and thank you all for joining today's session. So today we'll be focusing on a sorry, I think there is a technical snag. Okay, but in truth, the way the scientists write DNA hasn't changed much since the process was first developed in 1981. It's slow, laborious, and environmentally potentially hazardous. Commercially available DNA synthesis uses a process called as phosphoramidite chemistry, which relies on toxic flammable reagents that create hazardous waste. The process has important limitations. Most companies that build DNA this way top off at lengths about 100 to 150 base pairs, which isn't even as long as many genes. And the harsh chemistry involved in adding each A, D, C, T can also start to degrade the already written part as the molecule grows longer, right? This method also produces molecules that aren't compatible with water-based biology. That's acceptable if you want to use DNA as a form of data storage, but to be useful in biological applications, DNA made through this chemistry has to be put through additional processing, which essentially increases the cost. So the start to reaching critical milestones in DNA synthesis has already begun. And the ultimate aim is essentially to generate DNA which is longer, with fewer errors, or faster production, right? So today's session, which is co-hosted by Pramas Life Sciences and DNA Script, brings to you an important step in democratizing DNA writing, which is through DNA Script's EDS-based oligosynthesizer, the syntax. Having that at your disposal means that you no longer have to wait for weeks to get your oligos and have things in your control besides being, besides the piece of being able to keep your research information proprietary and lots of other advantages, right? Our speaker for today is Dr. Steven Quistad. I hope I'm pronouncing your name properly. He's a molecular microbiologist with over 14 years of combined research experience in both academic and biotech settings. He's performed his doctoral research in the laboratory of Forrest Ruhr in San Diego State University, where he investigated corals, virus, and the origins of immunity. For his postdoctoral studies, he switched viral systems, focusing on the impact of bacteriophages on microbial community function at the Marie Curie Research Fellow at ESPCI Paris, as a Marie Curie Research Fellow at ESPCI Paris. Steven joined DNA Script in 2019, where he currently works as an application scientist. As I mentioned before, the session is brought to you by Pramas Life Sciences, which is partnered with DNA Script to bring their products in India. And Pramas Life Sciences has been in the field of genomics and proteomics for the past, 1.5 decades, bringing to you cutting-edge technologies to enable your research. Without much ado, I'll just hand over the mic to Dr. Steven. All yours, Steven.
Great. Thank you so much for that great introduction to the talk today. So as mentioned, my name is Steven Quistad. So I'm a senior application scientist at DNA Script. So I've been with the company for about 3 years now under a variety of roles within the applications team. And I'm really excited to be here today to tell you a little bit about our enzymatic DNA synthesis, or EDS technology, which powers our printer called Syntax. So this is a bit where I'll be taking you today. So first, I'm going to tell you a little bit about EDS, or one more time, enzymatic DNA synthesis. So you'll be well acquainted with this acronym by the end of this talk. And that's our proprietary technology supporting the Syntax printer. Then I'm going to tell you a little bit about a collaboration that we had at the Institute Pasteur, where Syntax was actually placed in the field for a period of 1 year. And as one of my roles in the 3 or so years I've been at DNA Script, I was in charge of running this whole program. And so hopefully I can provide some unique insight and for sure questions at the end if anyone has any questions about how Syntax works in the real world. And finally, I'll go into a bit about how which projects were actually supported through Syntax. So going into some of the really cool experimental details. So before I start with Syntax and EDS, just a little bit about DNA Script. So starting back in March 2014, DNA Script was kind of founded with this idea that there's this bottleneck that exists right now in the life science industry. So our co-founders were working in the synthetic biology space, and they really realized the bottleneck in their experiments and the bottleneck of moving these projects forward quickly was really their ability to have these pieces of DNA, which are the foundation of the entire life sciences industry. So that was one of the founding principles in DNA Script in 2014, was figuring out a way to provide quick access to on-demand oligos. So that was back in 2014, and then over the first couple of years, we went from our first codon, 3 nucleotides, to 10 to 50, to announcing back in 2018 our first 200 nucleotide piece of DNA. And the beginning part was really that R&D into EDS, which led to the foundation in 2018 of developing Syntax, which is the world's first commercially available DNA printer powered by enzymatic DNA technology. So Syntax was launched back in June 2021, and that will be the focus of most of the talk today and the types of projects that Syntax is able to support. And the idea of what we want to support at DNA Script is really powering and accelerating this bio revolution. So if I go laser pointer, just one moment. Okay. So what we're looking at here, so this is Syntax. This is what it actually looks like in the field. And so we have 3 components I just want to point out. So here's our software interface. So this is a touch screen. And that's actually how you interact with the Syntax, how you load your print job, press print, and you can view the results. And that's custom made by DNA Script. Then what we have here, this door. So this door actually opens up like that. And inside, that's where your reagent racks are. So those go into the Syntax printer. And then the fluidics bring all of the reagents into this main case of the Syntax. And that's where all of the liquid handling happens and where the synthesis actually happens. So the Syntax printer is actually part of a 3-part Syntax platform. So we have our printer here, Syntax. And what Syntax provides is automated synthesis, purification, quantification, and normalization of your DNA. So what this means is essentially you have your digital sequences, which you input as a CSV file, which is transferred into a print job using our software. And then you press print and you come back and your oligos are normalized and desalted, ready for use in your experiments right away. So that printer is powered through our chemistry over here. And so what we have here are the different kits. So this is our first version of the kits that we had. So when we released it in the beginning, the kit was called 60 cycle 96 oligos. So what does that actually mean? So we provide enough reagents to print 1 plate of at 96 oligos at 60 nucleotides in length. However, you don't have to always print a 60-mer. You can also mix and match. So you can also print 2 plates of 30 mers or 3 plates of 20 mers, whatever your application actually requires. But there's enough reagents that are provided for 96 60 mers. And at the moment, we offer it in the 4 normal inks, so our standard A, T, C, and G. And so all of these guys go into the Syntax right here. And then here is our console software, which you can see on the screen here. And this is how you centrally manage all of your syntheses. You can share them, you can view results. You see the progress of your synthesis as it's in real time. So those are our three main points. We have our printer, we have our software, and we have our reagents. So these are the specs of what we support. So at the moment, Syntax prints in a 96 well format, printing at 200 to 300 picomoles per well. So that translates to about minimum of 5 micromolar final concentration. And then in terms of hands-on time, so less than 15 minutes to set up a run. So I just want to emphasize that one point about Syntax is it's extremely easy to use. So when I was over at the Institute Pasteur, and I would teach people who had never used Syntax how to launch a run, it would take them, I would usually spend about an hour explaining EDS technology, syntax, etc., and then show them how to set up the run. And then after that, about hour training session, it really takes about 15 minutes. You're just mixing some enzymes, pouring some different buffers, and you put your reagent rack right into the printer. So extremely easy to use. You really need a sub-minimal competency in molecular biology, even barely able to pipette, and you can still launch a run. And so how long does it actually take? So that depends on how long you're printing. So the longer your piece of DNA is, the longer it takes to print because you have to go through more cycles. So what we have kind of here is these two kind of use case scenarios. So first, if you're printing a plate of 20 mers, so let's say your standard size-ish of PCR primers. So the full runtime from print to your final desalted oligos, it's about 6 hours. So what does this actually mean in practice? So you could arrive to the lab in the morning time, set up your run, have your oligos ready to go for your PCRs by early afternoon, perform your PCRs, by late afternoon have results, and based on your results, you could redesign primers and print again, depending on how long you want to stay at the lab. You could set up a PCR later in the night, or you can just set up your run where your oligos are actually ready to use the next morning. So that's a really cool component of how Syntax is set up with our console software. So you can actually set up a run, and once the reagents are open, they're actually stable for an entire week at 4 degrees. And so this part of the Syntax here is refrigerated at 4 degrees. So you can choose your run to start at any time of the night so that you arrive in the lab at whatever time you want to, and your oligos are ready to use. So if we go, so it's about 6 hours for a plate of 20 mers, and then if we get up to the 60 mers, you're looking at about a 13-hour runtime. And how this one has done practically is it's basically an overnight run. So you would set up your run sometime late afternoon, and then whatever time you want to arrive at the lab, your oligos are ready the next morning. So then we also have what's called a customizable iDNA. So that I stands for initiator DNA, and I'll tell you about a project later that takes advantage of this really cool part of our technology that's completely unique to DNA script. So what iDNA is essentially it's these short pieces of DNA that our enzyme TDT actually adds nucleotides onto. So you can think of it, TDT, like most enzymes, likes to have something to grab onto first, and that's the function of this iDNA. And the really cool part about this is that if you do any type of application that uses a constant sequence, whether it's a library prep, which I'll tell you about later, or cloning, etc., we can actually create customizable iDNA where you have your piece of DNA that's a constant, and then we can print onto the variable region. And then your final piece of DNA will have that constant plus your variable. So this actually, because this iDNA can be up to 60, you can have a 60 nucleotide plus a 60 printed, and then you're looking at 120 mer. And so that's one of the really cool things I'll tell you about a little later. And then finally, as I mentioned, our 4 inks, A, T, C, and G, and fully automated. So sequences in, oligos out. And so one thing to point out, actually, it's really cool about this exact timing of this seminar. So at the moment, AGBT is happening in Florida, and a lot of our team is out there presenting. And we've actually just announced in a press release last Friday, all these specs have been updated. So now you're not looking at 200 to 300 picomole per well. We're up to over 600 picomole per well. So this comes out to be greater than or equal to about 7 micromolar. We're not doing only 60 mers, now we can go up to 80. And we're not just printing A, T, C, and G, we've now enabled fluorophores and biotin. So the timing of this seminar really ended up being perfect. So this is a press release that was released last Friday, if you want to go to our website and see the specs for yourself. And so this is the kind of system that we really want to enable. So as I mentioned at the beginning, The bottleneck in life science industry right here is this writing part, right? That was one of the founding principles of DNA script. So we think through syntax, we can close this loop and really accelerate the biorevolution. So our ability, of course, to read DNA today has become very cheap, and this writing is the bottleneck, which we hope syntax will move forward and support. So a bit about showing you how the world is today and the type of world we would like to create. So the DNA synthesis world exists as a service at the moment. So because DNA script is the first commercially available printer that uses enzymes, our EDS technology. What that means is that unless you have already directly interacted with DNA script, every piece of DNA that's synthetic that you've ever used and ordered from a vendor was made, what was mentioned in the beginning introduction, through what's called phosphoramidite chemistry. And if we look at how the industry actually is, so at the moment, DNA is a service. So you ask for your DNA and you get your DNA. Whether you're talking about single oligos or genes, you ask for it and then you get it in the mail. So this is a very centralized service that exists in a few big oligo houses. What we want to do with syntax is create a decentralized global model where you get your DNA when you want it. So as mentioned at the beginning, so this chemical approach, things haven't really changed much in about 40 years. A lot of harsh chemicals are used as well, and they're usually pretty complicated to use. You really need people who have a PhD in organic chemistry to run them correctly. If you compare that to our EDS technology, because we're working with enzymes, everything happens in an aqueous solution. So this creates a greener technology, and we don't use all of these harsh inorganic chemicals. We also have a lot less waste that's created. And so the DNA as a service model too is complex. So we have these transactions that involve multiple touch points. So what that means essentially is you ask for your DNA and maybe it arrives on time, maybe it doesn't. As I'm sure some of you must have experienced during the pandemic, for example, some people waited a very long time for their DNA to arrive as the global supply chain kind of collapsed of DNA. Even outside of the context of a pandemic, I can say from personal experience, DNA doesn't always arrive when you want it to. So through syntax, we're giving you the ability to make the DNA when you want, and then you can use it right away. And so kind of this complexity is also associated with these uncertainties where a supply chain might not always be dependable. However, in your lab, when you have your own printer, you can do it as you want. And so, of course, like slow versus fast. So DNA as a service, even if you get next day from some oligo providers, it doesn't always come next day. With the syntax, you have control and power over when you want to make your DNA. And I also have actually had this happen to me, where when you order your DNA, you start doing your experiments and things don't work well. And then it turns out they sent you the wrong piece of DNA in a tube. So that was extremely frustrating for me personally. I have other colleagues that have experienced this as well. So with syntax, if you have the wrong piece of DNA, it's because you did it wrong, but you have the power to make sure that you know exactly what is in your tube and what you've printed. So you don't have to worry about being sent the wrong tube of DNA, essentially. So here's the kind of ecosystem that we really want to support, where we're able to iterate faster through experimentation, where you're designing, you're printing, you're testing, you're repeating. So really increasing the iteration speed for these biological applications. So a bit about some applications that are enabled currently. And so what you see here on the bottom are these orange things with a check tested by partners. So what that means, well, after we had launched syntax, and especially before, we partner with a lot of different groups, pharmaceutical companies, academics, all across the board. And this partnership was set up where they asked for a piece of DNA, and we sent them what they wanted, and they did their experiments and shared their results. So we didn't tell them what to do or how to do it. They just put it into their normal workflow. So the point of all these four graphs here, you'll see anything that's in blue has an E, which stands for EDS. Anything that's in orange has a C, which stands for chemical synthesis. And the take home of all four of these is blue and orange are near each other. So whether you're looking at amplicon purity, standard curves made for cybergreen qPCR, and some amplicon targeting that I'll tell you a little bit about here, we see blue is near orange, blue is on top of orange, blue dots are near orange dots. And also we've done some gene editing and site-directed mutagenesis through our partners. So just to tell you a little bit more about the NGS application. So this actually is an application note that's also available on our website. So I invite you to check it out if this interests you. So what you're looking at is basically a SARS-CoV variant calling experiment. And this is the main take-home figure here. So what this green box here represents is the zone of what's considered good coverage in terms of an amplicon library. So if it's within the green zone, it's considered a well-covered amplicon. If it's in the white zone, it's considered a difficult amplicon. And we have orange, in this case, which is EDS, and then the two blues, CS1 and CS2, for chemical synthesis 1 and 2, so just two different oligo suppliers. And what we see here is all the dots are kind of near each other, is the take-home, and then these amplicons, which have super low coverage, are hard for us. They're hard for the chemical suppliers. They're just amplicons that don't cover well. Excuse me. And so that's kind of the take-home here, equivalency to chemical suppliers. So how about some future applications? So we've done, through our partners, we've tested hybrid capture using biotin. We've done some single molecule fish, which I'll show you some really cool data on in a little bit. Dual label probes, we've shown that can work. And then we've demonstrated genes up to 2 kb that have been assembled using EDS. So once again, what's really cool about this slide actually is as of last Friday, we can say that 3 of these 4 are no longer future. Syntax can do them today. So one of the things that we launched last Friday and at AGBT this week is hybrid capture, fluorescence, and the ability to make hydrolysis probes or dual label. So actually, even though this says future, as of right now, 3 of the 4 you can do today on Syntax. With gene assembly, we continue to work on that to be enabled in Syntax. However, at this moment, while you can do it with our oligos, it's not enabled on the Syntax. So that's a bit about Syntax in EDS and DNA script and where we've come from. So now I'm going to tell you a little bit about the collaboration we had over at the Institute Pasteur, which I was in charge of running. So this is a Syntax in the field. So this was our whole room dedicated to Syntax. This is the space it takes on a lab bench. Below in these fridges, so that's where the reagents, most of the reagents are stored. And then this tube that's coming down here goes into this waste container. And so there's also a -20 here. So in terms of volume of reagents, most things can be stored at room temperature or 4 degrees. And then at -20, we have our enzymes that are stored as well as our nucleotides. So the goal of this demo lab, which is over at Institute Pasteur, was really to see if we placed a Syntax, how would it be used and what would it be used for? So it was installed back in February 2021 for a period of 1 year. It was placed within the biomics unit of Marc Renaud, who's at Institute Pasteur. And over the course of about a year, we established 17 projects across 14 different labs. And I'll be telling you about some of those really cool projects in a few slides. So what was actually printed in terms of size range? So what you're looking at here on the x-axis, this is the oligo size in base pairs and the number of oligos within each size range. So you can see there's a big peak around 20 mers, so around PCR primer size, as well around 50 mers, which is part of a really interesting application involved with rRNA depletion, which I'll tell you about in a couple slides. And just a bit about these upper and lower limits. So this lower limit, so currently you can't print below 15 base pairs on a Syntax. And that actually doesn't have to do with necessarily the enzymatic DNA synthesis portion, but it turns out desalting really small oligos is really hard, and you end up losing most of your oligo. So while you can do the synthesis, once you do the cleanup and the desalting, you lose your oligo. So that's how we put this lower limit on the software. And then this upper limit, so as I mentioned previously, now we're up to 80 nucleotides. So this 60 doesn't actually represent anything that's limiting in the EDS technology. The specs, because we launched it back in June of 2021, we had to be quite conservative when we committed to those specs about a year before that, so in 2020. So we were being very conservative at the time, but we can print much longer. So we said on Friday, we can do 80 with the Syntax. In terms of EDS, we've printed up to 280 with actually longer pieces we've done internally. So EDS can go much further than this, but this is what's enabled today. So across the course of the year, we had 49 different runs on Syntax. We printed about almost 5,000 oligos. And this is the size distribution you're seeing. So within the applications, surprisingly, we didn't have much demand at Pasteur for oligos longer than 60. So of those 4,704 oligos, there was one person who wanted one oligo. There was 62 base pairs. So other than that, the people who approached us, they weren't necessarily aware that 60 nucleotides was the limit. However, no one came to us asking for more than 60. So that was, we kind of were expecting people to ask for longer ones, but at Pasteur, they did not. So that's the size of what we printed. What were they actually used for? So what you're looking at here is kind of just the distribution of the different applications and projects that were enabled on Syntax. So we see a big green chunk for mutant construction. So this was part of a Candida project that I'll have a few slides on in a little bit. And then we printed some fish probes. So the Pasteur Syntax actually was enabled to do fluorescent probes before we released it last Friday. So there were a lot of groups that were interested in doing some fish. And then the various applications we have here, it's just kind of your smorgasbord of general molecular biology applications. So we were, most of the projects were dominated by a few high volume oligo projects. In total, there are 25 different people that printed on Syntax and used Syntax oligos. And these are the distributions of their projects. And the general feedback we received from everybody is there were no observed differences in terms of how the oligos performed in their experiments. So now diving a bit more into the projects that we actually had. So starting off, first, highlighting our custom iDNA or initiator DNA capabilities. So this is completely unique to DNA Scripts technology. So what you're looking at in P5, P7. So for those of you that are familiar with Illumina sequencing, you might know about these two things, but if you're not, no problem. So P5, P7, essentially, if you've ever done any kind of library prep for Illumina sequencing, at some point you have to put a P5 and P7 onto your oligo. And why you need to do that is the P5 and P7 enable the piece of DNA to actually bind to the Illumina flow cell. So all kits will use a P5, P7 somewhere. However, P5, P7 being a constant, we can take advantage of the custom iDNA technology offered by DNA Script. And that's what you're looking at the workflow here. So what we do, so this is our iDNA here, our solid support that we have our P5, P7 onto here. And in this context, you're looking at an amplicon library that was being developed. So they wanted to do some multiplex PCR. So therefore, you have a lot of different forward and reverse primers that you need, but all of those need to have a P5, P7 for the library prep. So the orange part is that constant P5, P7. We then print the forward and reverse primers. And then through a series of PCR steps, we have our final piece of DNA. We have our amplicon generated through the multiplex PCR. We have what we printed with Syntax, which is in blue, the forward and reverse primers. And then we have our P5, P7, which actually came from the iDNA. So why is this interesting? Well, here is a schematic from the group of why they were interested in this in the first place. So on the left, we have the NebNext Ultra 2 kit. So that's a very well-used kit for Illumina library prep. And these are all the different steps that are involved. On the right, we have this two-step PCR kit, and that's the one that uses the P5, P7 sequence. And this big blue space in the red box are all these steps here that we get rid of. So we get rid of, in total, 3 different steps. So what we're looking at is less time for library prep and less reagents. So this group was specifically developing, or is specifically developing an HPV amplicon panel. And they did some preliminary experiments, and they were very happy with the results. So they actually bought some custom iDNA plates from us, and they plan to use that to develop their entire panel. So another project that was enabled looked at the spike protein from COVID. So this worked with the lab of Dr. Felix Ray, who's the head of the structural virology unit over at Pasteur. And on the left, you just have some crystal structures from some of his papers. So he's worked with mouse coronavirus, chikungunya, a variety of viruses. Just the thing to highlight here, so this paper came from 2016. So he was studying coronaviruses before the pandemic. The pandemic happened, and his whole lab switched to COVID, like many labs around the world. So the issue that they were running into is normally for them to get a gene they were just ordering from a commercial vendor. However, they were experiencing delays up to 2 months. So in the context of a global pandemic, where we're trying to quickly understand how the spike protein works and develop vaccines against it, 2 months can be a really long time. So this is one example of having the ability to print DNA in your lab, how you can accelerate your innovation. So not just for the sake of science, but for the sake of vaccine development and saving lives. So we developed a strategy with them that involved a multi-site directed mutagenesis kit and then next day syntax oligos. And so the study was divided into 2 parts. So there were phase 1 and phase 2. So in phase 1, we introduced mutations to optimize just the expression and stability of the spike protein. And in total, there were 10 different ones. And then we built upon phase 1 and phase 2 to add 7 additional mutants to make kappa. And for those of you that are familiar with the spike protein, we have highlighted, you'll maybe recognize some of these amino acids. So things that are in orange are the ones in phase 2, where we added kappa, and in phase 1 for expression and stability. And this is kind of the workflow of what we designed. So the original plasmid, it was sitting in this plasmid that was too big for the kit that we were going to use. So we had to pop it out of that plasmid and put it into a smaller one for mutagenesis. And in this phase 1 for the expression and stability, we added these 10 different mutations. And then we built upon that same plasmid in phase 2 to create kappa. And this was the really cool kit that we used for the workflow. So this was the Agilent Quick Change multi-site directed mutagenesis kit. And how this kit works, so we start up here where we have our double-stranded piece of DNA in a bacteria. So then we take out that double-stranded piece of DNA and we design primers that contain the mutation of choice. So why this kit's really cool is you can introduce 7 to 10 mutations at once with a single mutagenesis round. So we have our primers, which in our case are printed on syntax, and we have these little pink X's, which represent the different mutations. We then do a series of thermocycling, and then we have our piece of double-stranded DNA, where we have 1 strand in blue, which contains our mutations, and 1 strand in green, which is the complementary. So one thing that they don't have up here in this diagram, so this is a piece of DNA in green that came from a bacteria. So most pieces of DNA from a bacteria actually contain methyl groups. And that's what we take advantage down here. So you can see here, now they have an orange and green, there's methyl groups. However, this DNA that we made in blue was done in vitro, so there's no methyl groups. And this allows us to take advantage of the properties of an enzyme called DPN1, which only degrades methylated DNA using, it's a restriction enzyme. So then we get rid of all this methylated DNA, and we're left with our single-stranded piece of DNA that contains all of our mutations of choice, which is then transformed into competent cells for downstream processes. And so this is the workflow of the results that we got. So I have it divided into the phase 1 and phase 2. So things that you see in blue, you can think of as experimental design, and then things that are in orange squares are results that we had from the experiment. So starting in phase 1, the spike optimization, so that's for the expression and stability of the protein, and we're trying to get 10 different mutations. So out of the 12 clones that we sequenced, we had 2 different situations. So we had one clone, clone 8, where we had all 10 out of the 10 mutants. However, it also had a spurious mutation. Then we had clone 7, which had 9 out of the 10, and it was just missing 1 mutation. So rather than sequencing more clones, that was taking longer at that time. So we opted to just do another round of mutagenesis to find one perfect clone that just has that 1 additional mutation. And that's what we did here with the second round of mutagenesis, leading to our perfect clone with no spurious mutations that had all 10 mutations. So this was then, this was the end of phase 1. It was put back into the pipeline to then make kappa. So in kappa, we ran into the same situation actually. So we wanted 7 more mutations. Out of the 9 clones that we sequenced, we had 2 that had all 7, but they both had spurious mutations. Then we had 1 that had 6 out of the 7. And had, so rather than doing more sequencing, we opted to just do another round of mutagenesis, and we got our perfect clone. So that's the Spike project. So that one actually is available as an app node as well. So I invite you to go to our website to check that out. Now a bit about custom rRNA depletion. So if you remember from the slide earlier, where there was that big spike in oligos that were 50 mers in length, so that's for this specific project. So what you're looking at a schematic. So it turns out that most of the RNA inside your cell is consisting of what's called ribosomal RNA or rRNA. And that actually provides no information about what proteins are coded. So we need to get rid of it before we sequence. We could, of course, sequence everything that's in the cell, and then about 80 to 95% of that data will just be wasted. So we want to get rid of that ribosomal RNA before we do our library prep. So how we do it? We're looking at here total RNA. So in blue, the ribosomal RNA, which is the vast majority, and in green, the mRNA, which is the target of what we want to enrich for. So first, we develop our single-stranded DNA probes. So those are the pieces of DNA that are 50 mers, and that's where we use syntax. So then those DNA probes bind to the strand of our RNA, and then we take advantage of the unique properties of RNase H. So RNase H will degrade any piece of RNA that's bound to DNA. So after that, we are left with a bunch of probes, DNA, and our mRNA, which we care about. So now we can just get rid of all the DNA by adding a DNase, and we're left with our enriched pool of mRNA, which we then can go into library prep. So what's the interest of syntax? So currently there's rRNA depletion kits, but they don't really work well for all systems. So we have two different basically types of probe kits. There's one, the first type is a pool of probes. So there you get the whole depletion kit plus pre-printed probes. So if you happen to work in a model system, such as E. coli, if you're microbial, or mice, or human, if you're in the mammalian sector, you can get different kits for these. However, as it's a mixture of probes, you have a lot of stuff you don't need, and that actually decreases the efficiency of these kits. And if you happen to not work in a model system, then you're basically completely screwed. It doesn't work well at all because your organism of interest is not in that pool. So the solution to these is these custom probes, where this kit is called Ribo0 from Illumina, and these are designed and synthesized into a kit. So you can get whatever piece of probe you want. However, you have to ask for it and have it delivered. So at the moment, Illumina, you can send your organism of interest, and after about 2 weeks, Illumina will send you the sequences that you need to have, but then you still need to order them from a vendor. So that can take another 2 weeks. And then they arrive in about total time of about 4 weeks. So the lab that was working on this at the time, the total time from experimental design to doing the experiment was about a month. And so what they've done is it turns out the algorithm that Illumina uses is not very complicated to design these probes. So they created that in-house. And then of course with syntax, we can print the same day. So what they've done in the lab is taken this process from 1 month down to 2 days to have your results. So really a huge increase in speed. So that's kind of the basic principle and the interest. And this is what some of the data looks like. So what we're looking at here is the percent remaining of ribosomal RNA. So we want this number to be low. So this is after the sample has gone through the kit, and we want to have as little rRNA left, which will be end up that wasted data. In orange, we have the QIAGEN FastSelect kit. So that's one of those kits that provides a pool of probes. And then in blue, we have the Ribo0 kit from Illumina with our syntax oligos, DS for DNA script. And this was done in a species called Leptospira biflexa, which was previously known to be difficult to deplete rRNA, and it's used in the lab. And so what we see is about a 10-fold decrease in remaining ribosomal RNA when you take the custom approach and our DNA script oligos. And so these data actually are being written up now into a paper. So they've been very happy with the results. So now moving on to another project. So this project actually right now was an abstract sent in to AGBT by a Pasteur scientist that was accepted. And he's actually presenting a poster on this exact project right now at AGBT. And this also is an app note, which was just finished before AGBT and should be available on our website soon. So another one I invite you to go check out. So this particular user, he was waiting for about 3 months for his oligos to arrive. And he was just waiting for 20 mers, standard PCR primers. Syntax arrived, and he was extremely happy because he could hit the ground running, and syntax essentially replaced all oligos ordered from vendors. because he could do it as quickly as he wanted to. And the project he was working on was this Candida glabrata, which is a rising pathogen. And he basically was creating all these knockout kinases. So he was knocking out 96 different kinases, and these kinases is how they're resistant to antifungal compounds. So these 65 mutants were made using Thermo Fisher oligos, and then here Syntax arrived, and he switched completely and made the remaining 29 mutants using DNA script oligos. And he was really using Syntax in this very iterative fashion, where he would design his oligos at the beginning of the week, print them, perform his experiments based on the PCR results, redesign some oligos, reprint later in the week, and then just on repeat. So the idea here, after he created his mutant, he would perform these kinase assays and look for growth inhibition after you knocked out a certain gene, which would suggest that it's involved with antifungal resistance. And this is just to show a little bit of biology. So we have here are the 4 antifungal compounds he looked at. And everything here is in quadruplicate. So for example, here, 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 and here is the same mutant. So here and here, and 1, 2, 3, here. So what this means is that if we knock out the gene and then it doesn't grow anymore in the antifungal compound, that suggests that gene may have been involved with antifungal resistance. So in this case, the particular mutant in this block of 4, this gene that was knocked out could be involved with resistance to amphotericin B and caspofungin, and doesn't have any effect with fluconazole and voriconazole. And he's also writing this up as a paper as well. And this is just what the data looks like in the end. So in orange dots, you see our mutants that were created using chemical oligos. And in blue, we have ones created using EDS oligos. And the ratio at this 1 is the ratio to the mutant wild type. So things that are close to this ratio of 1, we knock out and don't see an effect, versus these things that are super low, we knock out and nothing grows. So those are candidate genes to continue investigating for antifungal resistance. So now just show a bit about fish in SME fish. So fish or fluorescence in situ hybridization is how you look for localization of an RNA molecule within a cell. And a different flavor of fish is what's called SME fish, or single molecule inexpensive fish. And the main difference between these two approaches is in fish, you have one probe that goes after one RNA target. In SME fish, you have a probe where the first part is target specific, and then you create a probe that is actually at a constant region, in this case, flat. So then you perform your hybridization, and you have a target specific portion, and then the flap specific. And this allows you to print only one probe. So you can have one probe with multiple RNA targets. And this is just what the results look like. So we're able to show that our oligos work in SME fish as well as normal fish. So this particular system, they're looking at viral infection of this DAV virus in the Drosophila system. What you're looking at is a gut of a Drosophila with nuclear stained DAPI. And these SME fish probes had ADO647, so that's the red part. And what these red parts represent are cells that are putatively infected with the DAV virus. And that's one thing they're investigating in the system, is which virus cell types in the gut are infected by DAV. And then they did some comparisons of SME fish and traditional fish, so just some more beautiful images. Here we have some more close-ups of these. So you can see these cells have very nice staining with DAPI, as well as ADO647, suggesting viral infection. And based on their analyses, they didn't see any difference with SME fish or fish. So just to kind of close up what I've shown you here today. So we've been able to support 14 projects across 12 different labs at Pasteur. And what we have been able to provide through Syntax in the real world is the speed, control, and dependability, which has enabled many different applications across ranges of molecular biology. And what we're trying to do with Syntax is fill in this gap in the bottleneck. So using the ability to write, to further accelerate and move forward the bio revolution. So with that, that's all I have for you today, and I'm more than happy to take any questions. Thank you for your time.
Thank you, Dr. Stephen. And I'm sure it was a really enlightening session for all of us present here today. and to actually figure out that, I mean, you don't really need to be dependent on service providers for providing you oligos, and nor do you need to share your proprietary information. So we have a couple of questions where, I mean, I'm sure you can add a lot of insight. The first is, what is the turnaround time for 2 to 3 kb of gene synthesis? While in your presentation, you did describe that at the moment, DNA Script has come out with 2 kits, one for 15 mer and one for 60 mer. But I also understand that oligos can be joined to create longer fragments and do gene synthesis. So would you like to shed some light on how much time would it typically take?
Yeah, sure. That's a great question. So the limiting factor actually there will be the kits that you use to do the assembly. So it's actually not necessarily how quickly you can print them. So depending on how long you want to make a gene, of course. It depends actually the kit you use that's on the market. So because we can print overnight, essentially, it's the limitation's not necessarily syntax, it's whatever assembly kit you're using to make your gene, if that makes sense.
Yeah, I mean, so Puneet, do you have any more questions on this? Like, do you want to take this question a little more and probably elaborate on it?
So elaborate on which aspect would you like?
Sorry, I didn't get you.
Oh, sorry. So elaborate on which aspect of the question would you like?
No. So I was asking this question to the person who had put up the original question,
Okay.
whether or not they want to, whether or not they want to elaborate the question, or, oh, I think he's okay with the answer.
Okay.
All right.
Well, I can actually
do you
one
have
more.
just to extend on that question, do you have like a time frame on which probably you'll come out with longer oligos? The capabilities of syntax coming up?
Yeah, so because we just launched 80 last week, the timeline for longer at this point, I don't know if we don't have a timeline at this moment, but as I mentioned earlier, we can print a lot longer. So at the moment, it's just the types of markets that we're trying to target in the beginning with syntax. And most of those markets in the beginning, the 80 nucleotides is actually sufficient. So at the moment, it's not on the very soon horizon, just because of where we're starting off. But in terms of long term, we definitely want to go longer.
Got it.
And actually, and just one more about the gene question that I can talk about a little bit more. So in terms of giving you a better feel for a timeline, so we're actually part of a collaboration with DARPA now. And the goal of this collaboration is essentially you have to be able to create a gene in the size of something that fits on a truck within a couple of days. And so we're part of this collaboration. So making a gene in a couple of days that's the goal of the project. So I can give you some numbers on that.
Okay, great. So there's another question which says, what is the quality level of the printed output? And I'm assuming, they're probably comparing.
Sorry, you cut out a little bit.
To the quality, and you've used and enzymatic DNA synthesis. So if you could just elaborate on it a little bit.
Yep.
And secondly, what is the capex cost and line? Because we have, Primas would be dealing with those prices. So the pricing, the commercials can be discussed offline, but I believe Stephen can comment on the quality level of the printed output as against the commercially available.
Yep, yeah, no problem, more than happy to. So currently our error rate is comparable to what you can buy on the market. We're better than some actually. So we're within the range of what you can buy today. So we have a coupling rate of 99.6%, which is also within the range of what's commercially available. And the data that we use to create those numbers is through NGS, essentially.
So Stephen, enzymatic DNA synthesis, like if you just, figure out what is happening in the cells, within the cells, like in vivo, compared to what is happening in vitro here, in vivo error rates are much lower, right? And, do you see a point where the error rates will be able to mimic the in vivo error rates, which is like really adequate?
Yeah, that's a great question. So within DNA Script, our enzyme engineering team has been kind of going after this problem from the beginning. So the TDT that we actually have in our system is extremely engineered. So it's not anything you find in the wild. So we're continuously trying to improve the TDT, and we are always releasing new chemistries and new mutants, essentially, that improve on this aspect. A challenge always happens as you get to super high efficiency, where it becomes you lose some things and you gain some others. So we already know some codons are more difficult than others. So maybe we get a mutant that's better at the ones that were hard, and then it's not as good as the ones that were easy before. So, and we're very much at this asymptote. However, we're continuously improving. We're taking different approaches for our enzyme engineering. So long term, absolutely. Timeline of getting there, I'm not sure.
Sure. And I'm sure a lot of people might want to actually understand a little more on the possibility of generating RNA oligos, because, RNA vaccines post-COVID have become the rage. So any plans of venturing out there?
Yes, we're extremely interested in that. We don't have any disclosed programs at the moment, but for sure, we're particularly interested in RNA because at the moment, the quality of RNA you actually can buy is very poor from commercial providers, very, very poor. And so even coming into the market to compete with them, it's not like DNA where what you order from oligo providers is high quality. So our RNA already exists poor in the market, so it's something we're very interested in developing, definitely.
I think if there are no more questions, are there any more questions from anybody in attendance? If not, then I think we'll just wrap up the session. Thank you so much, Stephen, for talking about the syntax system and the DNA script application around it. And we really hope we'll be able to have more sessions on this as the word spreads. And I'm sure more people will be interested, and we'll come back with more questions. And thank you so much again.
Great. Yeah, thank you so much for joining today. And feel free to reach out via email if any follow-up questions come up.